My research is in genetic pest management involving mosquitoes. So in genetic pest management for mosquitoes, you have two general approaches. One of the approaches is population suppression, and the other approach is population replacement. The approach that I'm taking is basically designing a system to push genes through a population and taking a wild population of mosquitoes that's able to transmit disease. And when we push these genes into the population, you'll end up with a population of mosquitoes that can no longer transmit disease. So to make transgenic mosquitoes, we actually spend a lot of time creating different constructs, which we basically take different pieces of DNA that we're interested in injecting into the mosquitoes that have a certain function, whether it's a lethal gene or an antipathogen or an effector gene. And after we have built these constructs, I will take mosquito embryos, these very, very tiny mosquito eggs, and fill these needles with the DNA constructs I've created and inject them into the embryos. Then 3 days later, I will hatch them in water. Once the ones that hatch become adults, I will cross them with wild-type mosquitoes, and then using our fluorescence microscope, I will be looking for different eye colors. We either use a green fluorescent protein or a red fluorescent protein, or sometimes blue. And we use that microscope to look at the fluorescent eyes and know if our functional genes have been taken up by looking at the physical conformation, which is the fluorescent marker. The end goal eventually would be to reduce the cases of disease that mosquito transmits. So those are really the big picture implications.